Okay, now we move on to 1944, where 1944, where the Second World War was full swing. It was going on in full swing. Then Avery, macleod, and maccarhy, three scientists got together and they performed an experiment that led to another big discovery, okay, or confirmation. We ended up the last video in Griffith finding out there was something from the S string that was converting the R string into a violent S string again. There was something in that heat-killed S string which was changing it, which was transforming it. Okay, and these three scientists, experimented. They did some experiments in order to find out what was the cause of this transformation. Now, before we go into that, let's just talk about this a little bit. RNAs, anything that ends with "ase" is an enzyme. Okay, and we know that enzymes have the ability to break down organic matter. So RNases are enzymes which break RNA. And enzymes are very, very specific. They work with very specific organic matter. DNases are enzymes which break DNA. And proteases are enzymes which break proteins. Okay? So with that as background, we go to the experiment, what they did. Okay? They took this simple technique and they treated the heat-killed S string with these different enzymes. So first, when they took the heat-killed S string and treated with proteases, transformation occurred, meaning it did nothing to that S string at all. Or that something which is present in the S string was not hampered or damaged by proteases. And secondly, they took RNases and treated with the S string. And after the experiment, they found out the transformation still occurred, meaning, again, that's something which is present in the S string was not touched by the RNases. And so the R string, the bacteria, turned into the violent S string. But when they mixed DNases in the third experiment with the heat-killed S string, transformation did not occur at all. So the R string bacteria remained R string at all. It remained R string. Therefore, it was acted upon by the body's immunity, and all these R string were killed. And therefore, transformation, the organism did not die. Meaning, transformation did not occur at all. So this led to the conclusion that DNA was the cause of transformation. And this was a big, big, big story in that time. Nobody believed that it could be DNA which was actually able to carry genetic information or the ability to transform. And they also wrote it in a way in which they suggested that only this bacteria, this particular bacteria, had its transformation power in DNA. And they even said, they even went another step forward. They even said genetic material of bacteria was DNA. Now that was a very, very strong statement. Not all believed. In fact, most people did not believe at all. Then, in 1952, another pair of scientists came to this conclusion. Hershey and Martha Chase. And Hershey-Chase experiment is also regarded as the classic experiments in biology, as it brought to, it answered a very big question with a very strong, emphatic yes. So in order to understand what they did, let's go on to this diagram. I've tried to draw a bacteria here. This bacteria is a very big one, okay? But I've just drawn a portion of it. This is the cytoplasm of the bacteria. This is the capsule, and we know that these are viruses. Okay, viruses have, most of the viruses have RNA as the genetic matter. Now, examples of certain viruses that you've already done in class 11 is tobacco mosaic virus and bacteriophage. So we are interested in the second one, bacteriophage, which was the one that they used in their experiments. So bacteriophage has a very simple structure. This virus, and all the viruses have a simple structure. It has a DNA covered by proteins. So all this green stuff that you see here is protein. It's a protein coat that protects the DNA. And the DNA core is inside. So all that the DNA contains is the information to make this protein. Okay? So this virus acts as a crystal, or it acts as a non-living organism when it does not come in contact with any living organism. But when it comes in contact with a bacteria, it injects its DNA, and it captures or kidnaps the DNA of the bacteria and makes it work for it in order to produce more viruses. Once all of it is used, the virus breaks apart and releases all these newly produced viruses. Okay, so that is what actually happens here. Now, in order to conduct the test, they actually used markers, and these are radioactive markers. So I've just done some glowing kind of stuff here, just to make you understand that it's radioactive. It can be easily traced. Okay, that's the basic logic behind it. So proteins are made of amino acids, and we understand that not all the amino acids have sulfur, but the most important thing is here that two amino acids, methionine and cysteine, which has sulfur. Okay, these two amino acids have sulfur in it. So, and methionine is the starting code. So, in order to copy the DNA, it needs a start code, which we shall be looking at in a later video. But just understand that this start code, or AUG, this coding is always present, needs to be present in the DNA. So, what I'm trying to get at is this: the proteins will have some amount of sulfur. Therefore, that protein was marked by, they inserted this radioactive sulfur in protein. And in DNA, we know that it has a phosphate group. In that, they use this phosphorus 32 as a marker. Now, let's say this protein coat has sulfur, and this DNA has phosphorus 32, sulfur 35, and I'm just going to write 35 here and 32 here. So they are marked now. So what they are trying to find is this: what is this that is injected into the bacteria, which actually captures this DNA and makes it work for that virus. Okay? So they also knew that the protein would always be outside the bacteria. And there's something called centrifugation. It's something like your washing machine when it dries up your wet clothes. It spins at a great speed, and it pumps out all the water. Centrifugation is spinning in high speed. So when bacteria and virus mixed in a sort of medium is spun like that, we find the bacteria will end up at the bottom. This is a test tube, and let's say the viruses on the top. Since they are very, very small, lighter in weight, and compared to bacteria which are bigger, they end up at the bottom. So this is a method of separating the virus from the bacteria, okay, centrifugation. So once these are marked, we call viruses, were allowed to infect the bacteria, and before the bacteria breaks apart, they took it into centrifugation and found when they used the protein, what we call the protein marker, they did not find anything. They did not find any radioactive particles in the bacteria at all, because all of it would stay. This protein would stay with the virus above here, okay, up here. So this bacteria will not have the radioactivity at all. And then they use these viruses, which were marked by this phosphorus 32. Once they injected its DNA there, then they begin to multiply that, replicate that, and then form new viruses. All of it would have this radioactive, part in it. So this bacteria is not yet broken apart. So now they took it for centrifugation and separated the virus from the bacteria, and they found that all the radioactive material that was used as a marker was present inside the bacteria, proving that it was the DNA which was actually the real cause of transformation. So this put a very strong stamp on DNA being the transforming agent, or DNA being the genetic matter. Okay? Right, this is it.